All right, so it's working. Anyone hear me? Am I?
Yep.
Okay.
I can hear you.
Cool. So I'd like to start out today with a wellness check. We've been through 2 weeks now. How's everyone doing? This is the final drop day also.
Good on my end.
You're good? No concerns?
Nope.
Okay. Anyone else?
No concerns.
So far, so good.
All right, everybody doing okay with quarantine stuff? Other classes going fine? But in particular, is this class making sense for you? If there are any parts of it that aren't making sense or you're unclear about, please ask.
I do have a question on the upcoming assignment.
Great.
So do you want us to, you have those two reference pages that you want us to start off on first, right?
Mm-hmm.
And then then going from there, we can spread out to see if there's any other material that we can use. Is that right?
Is this for the how many Ask My Seats?
Yeah.
Yeah. For that, yeah, I want you to use your Googling skills, okay? With the intent that you find peer-reviewed papers.
Right.
Yeah.
Is there a certain format that you want us to follow? MLA, MAA, or anything like that? Or
I don't care.
you don't care as long as we cite it and keep it the same way?
It could be a bunch of different ways. It doesn't matter. It doesn't matter.
Oh, okay.
They all have the same information. You know, they're easy enough to look up. Nowadays, you know, with the internet being kind of a thing, it doesn't really matter so much because all these journal articles have a DOI, a digital object identifier, and that's going to be the same no matter how you cite it, and that usually just comes in if you like download the citation from the site or whatever. So yeah, it doesn't matter to me. I just want you to get an answer and be able to kind of back up what you say.
Back up the finding.
The thing about that assignment is that I'm expecting different answers from different people. And that's fine. All right.
Okay, thank you.
Okay, we had a question earlier about the readings. So if I put Webster and Weber 8, that does mean all of chapter 8. If I put 8.1, that means all of 8.1, no matter how many subsections it has. If I put 8.1.2, that just means that particular several paragraphs. Okay. How is it going with your lab stuff? I've only got just a couple submissions so far for anything.
Yeah, what are we all supposed to have done up to this point with the lab, other than having our bread mold growing?
Yeah, have done is probably not the right word, but have started.
Yeah.
Have started your bread mold, getting it isolated onto a plate and getting pictures through a microscope, and have started capturing your chytrid in the pollen tube, and after today have started the Ascomycete stuff.
Okay, thank you.
Yeah.
Do you have dates for any of when we move in?
I'm sorry, you were really breaking up a bit.
Do you have
There you
any
go.
dates that you want all of these turned in?
No. I would like to, but it doesn't quite work that way because everyone's fungi are going to grow at different rates, that kind of thing. Maybe somebody's microscope order got delayed by Amazon, so I'm not going to have hard deadlines for any of these. The hard deadline for all lab assignments will be the end of semester.
Okay, thank you.
Yeah.
With taking pictures of the zygomycetes, do you want us to take those pictures directly from the fungi on the culture, or do you want us to pick some off of the culture and put on a slide?
Both are fine. You're going to get better results if you put it on a slide if you're looking at the sporangia, because you can just zoom in better and not have a bunch of bread in your way. But if you take like a macro style picture where you just kind of hold up the microscope to the moldy bread, that works. I don't know that you're going to get great photos that way. So definitely give it a shot on the slide to look at sporangia. What I'm really hoping you can kind of pick out is mycelium that doesn't have septa and some sporulating structures. The idea here is just to kind of get a feel for what it looks like in the real world, you know, aside from the pictures I have on my slides. They're usually kind of idealized and it doesn't always work that way.
Okay.
All right. For the pollen tube,
Yeah.
How long are we supposed to let that incubate?
3 days to a week is usually enough. What you're kind of looking for is a bit of a film on the top of the water. When that happens, you're definitely ready. You may find stuff before then. If you just wait 3 to 5 days, then it's probably okay to, you know, try to scrape some pollen out and look at it. You may find stuff. If not, if you're seeing pollen and no chytrids, then just wait a few more days.
Okay. And then a couple more things. So for the bread mold, we're looking for black mold, right?
Yeah.
All right. Because the pictures I sent you, I just have a ton of green and white stuff, and it's
Okay. The white stuff is probably going to turn black in due course.
I'm hoping it does because it's been a while. And I sent you pictures of the white has sporangia growing out of it. Is it still going to turn black even though it's got sporangia coming out?
How long ago were the sporangia popping up?
It's been about 3 days now.
Oh, yeah, they should be turning black. Maybe there are some weird species that don't turn black. They're not melanized. So maybe you've got that. That's fine. I don't care if the sporangia are black as long as they're sporangia.
Okay, so you're just looking for like pictures of sporangia?
Mm-hmm.
Okay, cool.
So the bread mold that we put in the augers, the zygomites,
Yeah.
what should we be looking for by the time we turn it in?
You're not turning that in until the end of the semester. That's going to be your culture, part of your culture collection.
Okay, so we wait till the very end of the semester for this.
Yeah,
one.
just tape it up, stick it in a sock drawer, and keep it away from your fruits and veggies.
Right. I've got it. I've got a lot of like cotton swabs in there right now.
Okay, cool.
It's all white, but on the base is all black and yeah.
Yeah.
Anyway, sorry. I just
So,
wanted
yeah,
to make sure
just hang on to that because we're going to try to get cultures of zygomycetes, ascomycetes, and basidiomycetes growing. And then you'll just take photos of those, just like a cell phone picture will be good enough just to like look at the gross colony morphology.
Okay.
Hey, Dr. Zahn, one quick question. I remember you had like a video posted about the bread culture. Is there other resources? I was just trying to look for them on the chytrids and the ascomycete lab stuff. Where do I find that at?
It's like videos. The chytrids, I didn't put any videos because it was just
That's
so
okay.
I
I don't always need videos.
Wait a week. And then for ascomycetes, we're going to kind of go through the culture portion of that lab today.
Oh, okay.
And for the find out how many Ascomycetes there are assignment, there's really no way to make a video about that.
Okay.
I guess I could, but we can just walk through it here also.
And what about with the chytrid? It sounded like you do something with water. I just didn't see. And I don't remember us talking about that, the process.
Yeah. So I put up instructions on the. I realized I didn't have instructions on the actual assignment on Canvas, so I added those to it.
Oh, okay. There is
essentially it's just add some water to bring it to about, I don't know, halfway, 3/4 full, and wait a week.
Okay.
Leave the cap loose so that oxygen can get in, and that's pretty much it. At that point, the chytrids that are floating around in the water are going to find the pollen grains and get really excited. Okay. So they get excited, they'll start growing and eating the pollen grains, and that's what I want pictures of.
Okay.
Chytrids.
Gotcha.
The other way to find chytrids that works really well is to go buy a frog at the pet store and stick it in the water and wait for it to die.
Oh, gosh, that's crazy.
Yeah, so we're going to stick with the pollen grains.
Appreciate it. Thank you.
Yeah. I see Hayden's got a hand up. Apparently, I've got a hand-raising function now. This is exciting.
I was sorry if this is a dumb question, but do you want us to wait until we have like black mold to move it from our bread onto the plate, or just as soon as we have mold, move it?
You can do either. So one of the things is if you just take some mold and stick it on there, there's a good chance that you're going to get more than one type of fungus growing. So the cleanest way is to pick off one black sporangium, stick that. That way you know you just got that one species.
Oh, okay.
Yeah.
So right now mine is just only white.
Yeah, that can work too. Maybe wait until you get sporangia going and move those.
Okay. Thank you.
The reason for that, a lot of lab work in mycology is just constant battle to keep cultures clean and pure. So what I've done is on your little petri dishes, I've actually mixed in antibiotics, and that'll help keep bacteria out. It doesn't work as well as it has in the past because of antibiotic resistance is kind of everywhere in the world now. So you may still get bacteria growing, but the idea is that we're going to try to give the fungi a competitive edge. Now, if you got a bunch of dense clouds of hyphae on your bread and you grab some and stick it on there, there's a decent chance that that's like 2 or 3 species all growing together. And so you'll have 2 or 3 species on your plate. So the ideal thing is to pull off a sporangium and stick that on there. So we're using the asexual spores from one organism. All right, Austin.
Yeah, I just had a couple questions. One was just kind of a nuts and bolts question. For getting discussion points for participation, does that just take place here in class, or do we have to? Is there like a discussion board? I wasn't able to find anything.
No, it's just talking
in class,
asking questions about the readings and that kind of thing.
Okay, awesome. And then I was just wondering, so my chytrid tube, my pollen tube, when I went to go put the water in it, I accidentally filled the whole tube up to the top.
It'll be okay.
Okay.
Yeah.
Thank you.
Benjamin's got a hand.
Yeah, I was wondering, once we've gotten the bread pollen into our auger and we've already submitted our pictures from the bread mold, do we still need to keep the bread itself with the mold on it, or do we need to?
No, as long as it's growing and you can tell it's like expanding in your petri dish, you're good to throw out that bread.
Okay, thanks.
I don't want to get
that.
Yeah. All right, Matthew.
Yeah, so just a quick question in terms of the chytrid assignment. I don't know if I'm remembering incorrectly, but I want to say that you said don't use all the pollen, like we had to save some for something else, or do we just put the water right in, and you don't need to have pollen for something else? I can't remember if I heard that or not.
Yeah, all the pollen is good for in this class is finding chytrids.
It's just for the chytrids.
It's just a bait. Yeah.
Oh, perfect. Okay, thank you.
Mm-hmm. All right, I see hands from Austin and Benjamin. Maybe they're still up.
Oh, you had to turn
Sorry.
it up.
All right. Well, let's talk about ascomycetes. All right, so this is our penultimate group that we'll be looking at. The slides aren't too long today, even though it's the biggest group of fungi. There's only so much depth we can go in. So this is still the getting to know the groups kind of portion of the class. We'll be talking more about all these groups as we look at their ecology and bioinformatics and stuff later in the semester, but are you ready? I will share my screen.
Looks like Matthew has a question.
Oh,
His hand up.
I think my hand is down.
it is down. It's
Sorry,
fine.
it was up on my screen for some reason. Then when you started talking, it went down.
We're all seeing things. It doesn't matter. It's okay. All right. So ascomycetes. This is the first group within our superclade Dicaria, right? The other one being basidiomycetes. Okay. Ascomycetes probably have the greatest diversity in terms of morphology and biochemistry of all the fungi. We've got pictured here in the middle is a cross-section drawing of little apothecium in a lichen, okay? And you can see they're pointing to an ascus here. This is where they get their name, the asci. Right? These little furry fairy cups are ascomycetes also. We're just going to kind of get to know a few groups right now, in particular the Saccharomycetes, Sorderiales, Alternaria, and Xylaria are two genera that we'll get to know. All right. So, Dicaria. Luckily, we're into the fungi that we actually have decent phylogenies for at this point. Up until now, the rule has been, oh yeah, but this is paraphyletic. This one's paraphyletic also. We have no idea how these evolve. But in Dicaria, we've got good, very strong evidence, solid phylogenetic trees with good bootstrap support that says that Dicaria is definitely monophyletic and contains basidiomycetes and ascomycetes. Okay. Dikaryotic is a genetic condition that a cell can have, and that's what this group is named after. Who can tell me what dikaryotic means?
So karyotype lists all of your chromosomes. So you'd have two sets of chromosomes, right?
Close. Karya actually refers to the nucleus.
Okay, so two sets of nucleus in a cell.
All right, so what about human adults? What's their karyotype?
2N?
2N, all right. So we've got two different sets of chromosomes hanging out in all of our nuclei, or they came from two different gametes. Pictured here in this generalized life cycle, you can see diploid. There's our 2N. Haploid, right? Those are our gametes. And then the weird thing is dikaryotic, which we shorthand write as N plus N in the fungi. There may be some other organisms, some eukaryotes that do this also, but none quite as well as the dikarya fungi, and that's why they get this name. What's happening here is, imagine a sperm hitting an egg, and it donates its nucleus to the egg. The first thing that happens in animals is the nuclei immediately merge. What do we call that process?
Like being a zygote, or no? Or fertilization?
Yeah, there's a fancy word that has karya in it. From your intro biology, karyogamy. Remember that one? Okay, so plasmogamy are two cells coming together. Karyogamy is two nuclei coming together. What the dikaryotic fungi do is delay karyogamy, sometimes for quite a while. Okay, so gametes get together, the cells fuse, the male donates a nucleus to the female cell, and then the nuclei don't actually fuse. They stay in their haploid state, but the two nuclei are still there in the same cell. So this is what's weird about the dikarya and why they have their name. And basidiomycetes and almost all ascomycetes have this stage, and it can be quite a long time, even years in some cases. So gametes come together, plasmogamy happens, okay? The cytoplasms fuse, but inside of each little cell here, pictured in this little cartoon of hyphae, you can see they've got little septa. We've got two nuclei inside each cell, and they haven't fused together. When they do fuse through karyogamy, then they become diploid. All right. So here's our ascomycetes, named after the sexual reproductive structure where meiosis happens, called an ascus, or a sac. Okay, so sometimes you'll hear these called the sac fungi. Okay. Their hyphae have septi. All right. So as they're growing, that polar cell growth, they're extending themselves from the tip. They begin to lay down little cell walls. Okay, those have pores in the middle. All right, but the pore is typically not big enough for nuclei to get through. It's just for nutrients and other bits of cytoplasm stuff, mitochondria, all that good stuff. So there's some movement of cytoplasm between these two cells, but through the septum, because it's got a little pore, but the nuclei don't move through it. Okay. Pictured up at the top right, that's an ascus. Okay. It has 8 cells in it. What are those from? What could give you 8 cells from a diploid cell?
It's just 2 stages of meiosis.
All right, so meiosis takes 1 nucleus and turns it into 4. And then you just double those. So the ascomycetes, almost all of them do meiosis immediately followed by a round of mitosis. Okay, so that's how we get this. Any spore found inside of an ascus is called an ascospore. Okay, and there can be a lot of different shapes, but typically it's all just a sac, and for the most part, they're all going to have 8 cells, which become spores inside of them. Okay, so on the bottom, you can see some of the diversity of sexual fruiting structures. All right, so an ascomycete will spend probably 90–95% of its life in asexual reproduction, okay, just cloning itself. In fact, a lot of ascomycetes, we've never found a sexual stage for them. We assume that they may still be capable of it, and we just haven't gotten them in the right mood. Who knows? Maybe they've actually lost the ability to undergo meiosis. Okay, but they still have genes for meiosis hanging out in their chromosomes, so they used to have sex at least. All right. So they can have naked acini just kind of sticking out of the fruiting body, or it can be enclosed in a bunch of cells called a cleistothecium, or it can be like partially open with a small little window called a parathecium, or it can be open like this in an apothecium. Right? So if we look at this structure right here, that little cup shape is an apothecium, right? And all along the inner surface of that cup are a bunch of acini sticking out, okay? Hundreds of thousands of them sticking out there.
How zoomed in is that picture? Is that like macro level, like eye?
Look, this one?
Yeah.
Yeah. So each of those is maybe the size of a penny.
Okay,
Yeah.
thanks.
They can get fairly large, like as big as like a fist, or they can be real tiny, just a couple millimeters across. All right. So here's the life cycle, real generalized, okay? Most of the reproduction cycle is in this asexual loop, okay? So we've got asexual reproduction happening most of the time. Those are called conidia. If you see the term conidia, it refers to a spore that is asexually produced in an ascomycete. Okay. All right, so we've got a couple different mating types here. They're growing. They can come together, and we get a big one and a little one. These are kind of the sexual structures. Ascogonium, okay, that's the female part, it's bigger, and the antheridium, that's the male part, okay. They come together, the male donates nuclei into the antheridium, and then they hang out without fusing, the nuclei do, until it's right before time to do meiosis, okay. So the ascocarp, which is the fruiting body of an ascomycete, okay, carp referring to the body, right? It'll form, and then meiosis happens right before, or karyogamy happens right before meiosis. So it's kind of like a last second thing. Right before they make their ascospores, that's the only time when karyogamy happens. Okay. Inside of the ascus, we get karyogamy, then meiosis, and then a second round, or a round of mitosis. So we go from 4 nuclei to 8. So that's 4 pairs of clones in there. So 2 of these spores are genetically identical to each other, right? Those germinate, you get haploid mycelium. They're happy, they live their life. When conditions get funky, they're running out of room, the weather changes, they get a flush of rain, then they may start doing sex if they're near a compatible mating type. Okay. So the key for dikarya is karyogamy only takes place right before meiosis. This is way different from animals where karyogamy happens instantly. You spend most of your life in a diploid stage, and then you undergo meiosis later in life. All right, questions about this life cycle? All right. Yeah?
Do we have an idea of like what triggers the fruiting body or karyogamy to happen? Is there any stimulus?
Bad conditions is kind of the rule of thumb.
Okay.
Yeah, so they're going to be happy if conditions are good, it makes sense to clone yourself. When you do sex, you're rolling the dice on your offspring, right? All that random assortment, crossing over, all that kind of stuff. You don't know what your offspring are going to be like exactly if you go through meiosis, okay? Which can be a good thing. If the environment's changing, you want to roll the dice. It's not good for you right now, so go for it. But if things are good, it doesn't make sense to do that. At least that's the way that fungi kind of see things. Yeah. So running out of food, the onset of winter, like a change in the climate, that kind of stuff, that can trigger it. this is why we get so many mushrooms in the fall, if that makes sense, right? Things become a bit wetter, the temperatures are dropping, so it's time to roll the dice with meiosis and overwinter if possible. Okay, so this is just showing one type of fruiting body. What kind is it?
Forgot the word, but it's the open one.
Apothecium. Yeah, kind of looks like open. Apothecium. The other two are Cleistothecium, which is closed, and Perithecium, which is almost closed. So we'll look at another Apothecium right here. Okay. The reproductive, sexually fertile part of Ascomycetes and Basidiomycetes is called a hymenium. Okay. Hymenium just refers to the surface of the mushroom body that is going to go through meiosis. Okay, it's the only part that can go through meiosis. So you'll see that hymenium term. It just refers to the sexually fertile parts of a fruiting body. Okay. All right, so let's take a look kind of up close, starting here in this green box on the bottom. Okay, we've got our two mating types. They fuse through plasmogamy, and the antheridium is going to donate some nuclei to the ascogonium. Okay. Now it's dikaryotic, and being dikaryotic actually stimulates a lot of changes in a fungus. Okay. It can't make this apothecium up here unless it's dikaryotic. All right. It really needs to have those multiple nuclei per cell. All right. So you can see them pictured here, the two different sources of the nuclei. They're hanging out, and it does this weird little like folding over thing in order to keep everything to where there's two nuclei per cell. Okay, we call it a crozier, which just means hook, kind of like a shepherd's crook. Okay, so it kind of folds over on itself, lays down some cell wall there, and so we've got two nuclei. Okay, then this one wraps back around and they fuse, so we've got two nuclei there, and then right before meiosis they join together, blue and red make purple, so now we're diploid. And we're diploid for a total of, I don't know, 5 minutes or something in fungi, okay, or in our dikarya. So immediately after that, meiosis begins. Okay, so we get meiosis. There's our 4 nuclei, then a round of mitosis, which makes them 8, and we've got our ascus. Okay, those ascospores then get either passively or forcibly ejected from the ascus. A lot of times this kind of pops open like a cork and you send them out, and the process begins again. Okay. So we've got over here on the right the little GIF kind of showing how that happens. It's not, unfortunately, showing this little hook action. I cannot find a little animation of that, and I'm too bad of an artist to make my own, but I may try. Okay. On the left, we're looking at an apothecium kind of zoomed in here, and then zoomed in even further on the asci, okay, and the plural for ascus. Okay, so you can see this kind of structure here. We've got all these cells down in the bottom that we call the medulla, and that's only important if you see that term, okay. They are not fertile. They're not dikaryotic, or they are dikaryotic, but they're not diploid. Diploidy only happens right before the ascus is formed and meiosis happens. Okay. Is this making sense? It's kind of hard to demonstrate this without having microscopes handy, but we do have cultures of this that you'll look at. Okay. What kind of fruiting body is this in the top right?
The para version.
Yeah, perithecium. Okay. So our terms so far: ascocarp. That's macrostructure. These can typically be seen by the naked eye. They can be kind of tiny and just look like a little speck, but you can still see them. We've got the ascus, plural is asci. Those are the sacs that house the sexually produced spores. Ascospore is just a spore that was produced via meiosis in an ascus. And the hymenium, that's the fertile surface where the sexy business happens. Okay. Now over here we've got antheridium and ascogonium. Those should make sense to you, like anthers from plant parts. Okay, they're the male parts. They donate to the ascogonium, and that's just the thing that houses the ascus making supplies. All right. And then finally, conidium is asexual spore in ascomycetes. All right, so we've got some actual light micrographs of some of this stuff here. Okay, so we'll start at A, which is confusingly placed here. All right, this is a spore germinating little growth tube. All right, so that's a hypha coming out of that spore. Haploid or diploid? Or dikaryotic?
Wouldn't that be a haploid?
Haploid, yep. All right, so we've got a haploid mycelium starting to grow. Okay. Over here in B, okay, we've got the mycelium developing. So we've got a bunch of branches off of this original one. That's still haploid. Okay. And then in C, parts 1 and 2, we've got antheridial elements and ascogonium. The ascogonium just looks like a bunch of kind of swelled up cells. That's fine. So these are kind of the sexual organs for the haploid life cycle. They come together in part D here, which is kind of tough to see, but I'll try to outline the ascogonium for you. It's got a big cell here, kind of grows up and around. All right, so this has been fertilized. It's starting to expand. The nuclei are multiplying through mitosis, so it's growing and swelling. And then up here in E, we're starting to form an ascocarp. Okay, so we've got our ascogonium right in the middle, and that develops or signals some changes in the hyphae around it, and they say, okay, it's time to make a fruiting body. Okay, so they start growing. We've got ascogonious hyphae. You can see some of them crooking over. You can't really see nuclei in this photo, but this one right here is starting to make that little crozier, which means that it's beginning to segment itself and keep 2 different nuclei per cell. Okay. And then in G, these are young asci coming off of the hyphae that are making the asci. Okay, so these ascogenous hyphae, the things that make asci, they're kind of buried in the blur here, but you can see the asci developing. All right, so there's an ascus in H, and then in I and J, you can see developed asci. Okay, so after meiosis. All right, so this life cycle is a bit different from what we might be used to. The finished product, so to speak, here in K is, you can see that this is actually under a dissecting scope, so these are pretty tiny. Okay, but these are apothecia, and the little black fluff are developed asci on there, getting ready to shed their sexually produced spores. Questions about this life cycle? Nothing? All right, y'all are catching on to this stuff way quicker than I did.
The graph that you had before explained it pretty well.
Okay. I wanted to show you this because this is what real life looks like. It's not always as nice and clear as the cartoon version. It can get quite confusing if you don't know what you're looking at. You look under a microscope and see this shape in D here. You may be going, what is that? Okay. So I wanted to show you the confusing real version of what you see, because you are going to look at these pretty soon. All right. So we got 3 major groups of ascomycetes. The Taphrino mycota, Okay, the Saccharomycota and the Physiomycota. I guess these should technically be Mycotina since they're subphyla. All right. The Taphrinoes are now considered to be basal to the other groups, and you're going to see a different story in your textbook. Okay. These used to be called the hemiascomycetes, and that's what the textbook still says. Okay. That's because we've refined further the placement of the Saccharomycete yeasts in there. Okay, so we've got our Taphrino-mycota pictured here in kind of orange. Okay, they're basal. We've got our Saccharomycotina, which are generally almost all yeasts. Okay, and we've got the Physiomycotina, and these are kind of the molds and even some mushroom-forming things. You've heard of morel mushrooms, no doubt. Those are here in Physiomycotina. All right, so the Taphrinomacetes, you'll see them in your textbook called hemiascomycetes. These contain a bunch of weird things. Dimorphic stuff tends to hang out here. Dimorphic meaning that they grow as yeast sometimes and have hyphae at other times. We've got human pathogens in there. These weird fission yeasts that don't even bud, they just kind of split like bacteria. Okay, but it's a relatively small group and somewhat understudied. Saccharomycetes contain most of the budding yeasts. Okay, and some of these can still do hyphal growth. And then the Physiomycotina, named after the fact that a lot of them respond to fire, Physizo, okay, are the charismatic ascomycetes, if such a thing can be said to exist. Okay. So here's our Taphrinomacetes up at the top. Okay, you probably have seen these on a rotting log before. These little like jelly fungi looking things. Okay. Really important plant pathogens live in this group. The Schizosaccharomycetales are a really important group. The fission yeast, okay, they have split personalities. Okay. And then the Saccharomycetes, there's actually a bunch, but they get one little branch here. You can see how many groups there are, and this is at a family level. But here they are. These are important for a lot of reasons, but they're famous for being the #1 model organism for genetics of all time is brewer's yeast. Okay, so that's Saccharomyces cerevisiae, and that's also bread yeast. So pretty important microbe, first microbe to be domesticated, first eukaryote to have its entire genome sequenced, and it's really easy to work with. And its genetics are well understood, and so we use it as a great genetic model. And then the Physiomycotina, right down here. Okay, huge diversity in form for the Physiomycetes. Okay, we've got things like Alternaria, it's an important plant pathogen. We've got our little fairy cups. We've got tuber. What's the word for this? Truffle. Yeah. So black truffles. So important culinary stuff in here, and then of course the lichens. All of those are in a single group called the Lecanoromycetes down here, the lichenized fungi. What is a lichen?
Is it a cross between a moss and a fungus?
Like a mad scientist went to the lab and forced them to reproduce together, like a liger or something?
Yeah.
Then no.
Then no.
You're kind of on the right track.
Is it like a really basic version of a fungus?
Not necessarily. They're pretty highly derived. I mean, they're down here in the ascomycetes. They're not like chytrids or anything.
They have symbiosis with algae, right?
Symbiosis, yeah. So it's a mutualistic arrangement. So it's not a cross necessarily, because that implies like sexual reproduction. They can't do that. You can't like mate a fungus with an algae or a moss. But in essence, it is fungi that have learned how to farm green stuff. Okay. and it only is found in a monophyletic group, the Licanoromycetes. Okay, so these are ascomycetes that essentially depend on some green stuff to get their food. Okay, so they've, I don't know if enslaved is the right word because they both benefit from this, but they farm either green algae or cyanobacteria. So the green stuff lives within the fungal tissues. It still gets sunlight. It takes CO2 out of the air. It makes sugar. That feeds the fungus. And what does the green stuff get out of the fungus?
Protection?
Yeah, protection. It keeps them from drying out. It gives them a nice, warm, safe place to live. And it also is capable of getting some nutrients from like minerals and stuff, especially from rocks, that kind of thing. All right, so it's a nice little arrangement. We'll talk more about them when we look at symbioses.
Hey, Dr. Zahn, what was the
Yeah.
name of the one that was a really good model organism for, like, you said, like the best for genetics?
Saccharomycetes.
Saccharomyces. Okay.
Yeah, so Saccharomycotina, the fungus that likes to eat sugar is kind of what Saccharomycotina means. That's because it's really good at taking sugar and making useful things for humans. All right. The reason we have bread before people figured out baking soda and like Irish soda bread is you grind up some grain, you mix in some water and some Saccharomycete yeast, and what's it do?
It eats the sugar and turns it into something we like to eat.
In particular for bread, anyone bake? If you don't put yeast in, your bread doesn't rise. It's
Yeah, that
responsible.
turns
putting-
into a really interesting experiment if you ever don't put the yeast in. Even better if you don't put salt in, then it just tastes amazingly terrible. It doesn't take
Yeah.
much salt, but it needs it.
It does. If you don't put yeast in, you get crackers or hardtack. Yeah. The yeast is the leavening agent. That's what makes it rise. And so it's feeding on some of the starches and sugars that are present in your flour. And it poops out ethanol and carbon dioxide. Now, most of the ethanol gets evaporated when you bake the bread. Okay. Some of it's still trapped in there, but the main thing we really want from it, we don't want to like get drunk from eating bread, right? We want the bread to be soft and fluffy and easier
Whoa.
to digest.
Speak
And
for yourself.
Okay. That would be a pretty rough experience, I expect, to get drunk on bread, but okay. CO2. So biochemically, it's working just like an animal. It takes in sugar, burns it, and the byproduct is CO2. Right? And so that's what the gas bubbles are that make your bread rise. Right? So that's why we started using this as a model organism, is because like human civilization depends on it. This is, historians put this down as probably one of the biggest reasons why we stopped hunter-gathering and started laying down roots and doing agriculture, was because we enjoyed getting drunk, and you have to have a lot of grain and you can't be moving around for that. And so archaeologists have found the oldest pottery and stuff actually was used for making beer, not for making bread. So beer turned up at least a couple thousand years before bread did. And that's just grain plus water, let it sit out for a while until it starts bubbling. All right. These yeasts are all over the place. They're just floating around in the air constantly. Their genomes are relatively simple, and so it was our first target for getting a complete eukaryotic genome, because it's a relatively small genome, and then they're really easy to work with in the lab. So genetically, we can easily do mating type crosses, look at meiosis, all that good stuff. All right. So looking at these fungi, the question, especially when you look at lichens, is what is a species? Okay. So I would like all of you to define a species and just yell it out when you've got yourself a definition. Because lichens, they depend on another species too. That thing you see on the tree growing there, that lichen, it's actually at least 2, possibly 3 different species all living closely together in a symbiosis. Well, I use species in my definition of species. That's kind of circular. It doesn't make sense. You can see the problem. So come up with your definition of species for me.
I guess I'll go first. Mine's, I guess, more traditional. I when I think of species, I think of more of like the biological species concept, where they have to be able to interbreed.
Okay.
And then if I could choose, I'd like to back that data up with genetics after, I guess.
Okay.
Confirm.
So you would go with if they can interbreed and produce viable offspring.
Yep.
Then that kind of counts as a species for you.
Well, and then maybe you can follow that up with genetics with and like pick out a few sequences that, I don't know, Hox genes, whatever, something that's conserved that you're trying to look for.
Okay.
It's the same between them.
All right.
I'd also try and fit in somewhere along with his definition that they fulfill a niche in their area, because multiple organisms can have the same niche, but they usually outcompete one another in a specific, environment.
Okay.
And that's usually where a species comes from, as it's the dominant in that specific niche.
Okay, so you're going with the kind of ecological concept where maybe define a species as in a given area, the things that are doing X, Y, Z, they would all be the same species.
Yeah, well, yeah.
But X, Y, Z being actually like thousands of traits, I expect. Instead of just
probably.
Yeah, okay. Anyone else? These work okay, especially the evolutionary one, maybe. The biological species concept kind of breaks down quickly. It was developed for animals. What's a species of bacteria?
Isn't that a whole mess, because the Linnaean system doesn't even work well for naming bacteria, so we're having trouble identifying species?
Well, yeah, I mean, maybe the Linnaean breaks down, maybe it doesn't. Kind of the degree of which it works okay depends on who you ask. Most bacteriologists probably say it doesn't work very well. But one thing that bacteria don't do is mate, right? No such thing as a diploid bacteria. And so the biological species concept definitely doesn't work for bacteria, because nothing is ever breeding, and so you can't test whether breeding works or not.
I suppose for the for bacteria, then I would look at it more I would look at their genome, just not their genetics purely.
I think that's where people have gone in bacteria for sure, is kind of this phylogenetic species concept. So it gets tricky, of course. Where do you put the cutoff? If you're 99% similar genetically, do you count as a species, or is it 97, or is it 99.5%? And so debates rage on and on in there, because nothing quite seems to work. We have the same sorts of problems in fungi, especially ascomycetes, because for the most part, and in some quote-unquote species, for the all part, there's no breeding happening, right? These tend to be asexual. When we do find sex in them, it's kind of the exception almost. So if you spend 99% of your life in this anamorphic, asexual stage, how do you define that as a species? The biological species concept doesn't work. Ecological, maybe even not so much, because things can look different. They can have different looking conidia here, but be filling, as far as we can tell, the exact same niche. And so they're probably not filling the exact same niche, but to the best of our measuring abilities, they are. And so that kind of, at least by default of we don't know what we're doing, it throws out the ecological species concept also. And then we got the phylogenetic species concept, and it seems like the world of mycology is kind of moving towards that also, because there's so many exceptions to the biological species concept. This became a problem, well, it started out as a problem, I guess, is more appropriate, because this is Aspergillus, very common fungus, okay? It's got distinct conidia that look like this. If you really want to tell it apart morphologically, you have to get in with an electron microscope and look at like the shape of the spores and the ornamentation patterns on the spores. Other than that, they all kind of look alike. This is the shape of their conidia, okay? Their spores look alike under a light microscope. So this is Aspergillus. We call that the mitosporic state because the spores are made via mitosis, okay? This is Neosartoria, which we saw often, and the Linnaean terms seem to work out for it. We could do mating crosses with it, but for quite a while, nobody knew that these were the same organism. Okay, so they got two different names. That's exciting, all right? So Neosartoria and Aspergillus are the same species, if species means anything, okay? One is just the asexual form, one is the sexual form. So what's the problem here?
Just about everything, it's the same organism.
Yeah, it's horrible. So if you're talking about stuff and let's say you find a sick patient with an eye disease and you isolate the organism, it turns out to be a fungus, and you go and sequence it and it comes back as Neosartoria. You're a doctor, you're a medical professional, you're worried about this, so you run that through a little literature search. Neosartoria, has this ever been found to infect human beings before? Let's see if we can find case reports so we can know how to treat this. And it comes back nothing. What do you do?
Well, since they probably aren't specializing in fungus, they're just like, well, let's look for other causes for this.
Yep. And that has happened repeatedly in medical mycology is, well, until very, very recently, like the past couple of years, most medical programs dedicate 1 day of lectures to mycology. That's it. And so that can kind of get lost in your training, right? And you've got several years of med school, and then you go and do your clinical rotations, and guess what? You forgot all about the fact that anamorphs and teleomorphs exist. All the mycology research up to that point was using this name Aspergillus, which is actually a fairly common human pathogen. And so you just missed out on a body of literature that could help you save this patient's eyeball because it has two different names. And so you'll see this with fungi. There'll be papers reporting some new occurrence of Neosartoria in a human. And, well, 10 years ago, the same thing was noticed. They just called it Aspergillus. All right. So this is a major problem. I hate it. But it's something you got to deal with as a mycologist. So if you do find yourself in medical school, please remember My dog is really snorting it up down there. Please remember that a lot of fungi have two names in the literature, which is really horrible. Okay. From your text, this is just a small little sampling of some of the problem here. The left-hand column is the teleomorph name, right hand is anamorph. So sexual, asexual. Okay. Neurospora. This is a very famous, another model organism. This is where we found like how genes are linked in the chromosome and all that good stuff. Chrysonilia is the anamorph name. Okay. Nodulasporium is the anamorph of all of these different genera of teleomorphs in the Xylariales. So Xylaria, Daldinia, they get the same genus over here in the anamorph, different genera over here in the teleomorph. This super sucks for mycologists.
So have we just continued this trend?
Yeah.
Or why?
It's because this is really stupid. It's because we are bound by the Linnaean nomenclatural rules of the Botanical Society. Back when people still thought that mushrooms were plants.
Why? Why are we bound by that stuff?
I don't know. I'm sorry. I went to one taxonomic session at a conference, and one was enough for me. It was old guys yelling at each other around a round table about names and whether or not we should keep these names or not. Like screaming. They were angry, and it was just contentious old people yelling at each other about stupid things. That was my impression. I feel like having two names for something for the same organism is a major problem, given that these are important human pathogens, but even more important plant pathogens. And so if you're looking at some fungal outbreak that's sweeping through the Pacific Northwest, wiping out crops, and people are talking about it with different names, wires get crossed and poor decisions get made. So I and a lot of mycologists find this absolutely mind-boggling that we're still doing this, but we are. So what's the solution?
You wait 20 years for those old guys to kick it and then go in and change everything.
Well, they're the ones training the next batch of mycologists.
Yeah.
So
But
that may not be long enough.
But you're training us.
Credit, right? Everybody wants the credit for their past discoveries, and part of it's the history that's involved. Like we need to know that history on how we, came to the current knowledge that we have. So it's kind of hard to move forward with a new system and honor everything that's happened before.
And it's not even just a matter of credit, but when you look at old papers, right? So there's this huge body of literature, right? Literally thousands of papers on Neurospora going back, into the 1950s. And if we suddenly start calling it Chrysonilia, we'd have to change each one of those publications also. And that's quite a process. And so the body of science that's been building off of learning about this one named fungus kind of gets thrown out at that point. It's actually a difficult conundrum we're in. And it's just historical accident, a mistake that people made. We thought these were plants, so we stuck them in the botanical nomenclature, and now we're kind of stuck there, and we've done so much work on it using these quote-unquote wrong names that we're kind of between a rock and a hard place, I guess. For me, I don't even like using fungal names, and this is why I am bad at identifying fungi and naming them. And I feel bad for the biology department at UVU because they hired a mycologist thinking that I'd show up and be able to look at a fungus and tell them the name of it. And then I get here and tell them that I don't believe in fungus names, and somehow they still hired me. So I don't know. I don't know what to do about it. For me, I just tend to go with more of a phylogenetic species concept for fungi. I think we're at the point now where sequencing is easy enough and cheap enough that we can kind of get away with that phylogenetic species concept. But if I do find something, a sequence of interest in a sample, in some of my research, and oh hey, this one is really affected by, you know, this human phenotype, they tend to have more of this fungus, then I have to still go and look it up, but I have to be cognizant of the fact that it might be under the literature under a couple different names, possibly more. So you look at hypoxylon, it's Nodulosporium, but also Geniculosporium. Okay.
So essentially what you're saying is that if we can't figure out the name, we need to realize that there might be two, and then even at that point, we just refer to what Batman's doing right now.
Yes. Now, there is a website that's trying to get around this problem. It's called Mycobank, and let me see if I can take you there. Mycobank. It's been having connectivity issues recently. Okay. So we can just do a search, basic search on names here. Taxon name contains Aspergillus. Oh. So let's say you're doing your research and you've sequenced some sample from a patient or a sick plant or something. You want to know what it is. You would come to Mycobank and let's say, oh, okay. This is Aspergillus acanthosporus. That's what the DNA is telling us. So I click on that. This is all still kind of crazy. And I come down here and boom, teleomorph synonyms. Okay, Hemicarpentales, Neocarpentales, and Neocarpentales. So we've got two different names, or three, including the Aspergillus acanthosporum. It's also sometimes in literature called Hemicarpentales acanthosporum, and sometimes Neocarpentales acanthosporum. So I would then do a literature search on all three of those names and like see if my patient or my plant has reports on it in the past. Look for old scientific literature that has that stuff going on. All right. Okay. Not all ascomycetes have this problem because not all of them have been identified with the teleomorph stage. Okay. And that's just because some of them either can't have sex anymore or we haven't figured out how to get them to have sex in a lab so we can prove that the teleomorph and anamorph are the same thing. Okay. Sexual reproductive structures are easier to tell apart, but we've got some characters for the asexual structures. So down here, these are sexual structures, these two in the bottom. These are all asexual structures, so the anamorph diversity that we can see. So if you see this shape here, you're pretty certain that you've got some Aspergillus on your hands, or at least well, that's one name for it, at least. Okay. Cladosporium, it kind of looks like a little tree, clado for tree. Alternaria here. These all look pretty unique and different under a microscope, so it's not an impossible task to get a microscope out and key these out with a dichotomous key and see what quote-unquote species you have, but it can be kind of tricky. So this is how the DNA databases have been made, right? So I'm standing on the shoulder of giants in this case. I like to take a sample, grind it up, and for $4, sequence pieces of its genome and match that up to a database, and it says, oh, you've got Alternaria acanthosphorium. Great. Okay. So that's how I get fungal names. But in order to put a name to that sequence, we had to have somebody grow it in the lab, identify it morphologically, call it a species, and then update the database and say, this is what the genome of this one looks like. Okay, so there's still work to be done in this case, making our databases better. Okay. So in fungi, when we're trying to talk about species, what nowadays people are talking about is not the biological species concept. They're talking about molecular phylogenetic species concept backed up by secondarily morphological species concept. So if it looks exactly the same under light and electron microscope and the genomes match up, then we say, okay, it's the same thing. All right. Keeping in mind that it may have 2 or 3 names. Okay. So if we can't find a teleomorph state, we're stuck with the anamorph, the asexual. And this is 90% of all ascomycetes spend their time in this mitosporic stage anyhow. All right, so we're often looking at this under a microscope. It's kind of the exception to find asci being formed and all that stuff. Asexual is the default. Okay, so we've got all these different conidial structures. Okay, we've got spores that all look very different from each other, and so that's how we identify stuff. Now, you can make these mitospores in a number of ways. The 2 main ones are blastic and thallic, pictured here. Blastic is it's blasting out. Okay, so it's building one bud on the bottom that pushes out. It builds a new bud and it pushes out. And then the thallic is where you just take your body, divide it up into some segments, and let it break off. Okay. Both of these make valid mitospores or conidiospores. All right. Important thing to remember is that this is just an asexual haploid hypha that's being modified in some way. That's what a spore is. Okay. Questions about that so far? All right. So let's watch some fruit rotting. This is actually zygomycete that we're seeing here, but it's a good little video. This is showing, we kind of talked about this already, but why asexual reproduction is still selected for evolutionarily, why it's advantageous. Okay, if you are good at a given environment, it does not make sense to roll the dice with your genetics and reproduce that way. It's better off if you just clone yourself, okay, because you can take over the food source faster. Okay. Only when you run out of food or you get environmental conditions that say winter's coming or something, that's when it's good to try meiosis. Okay, so we're going to look at 4 groups just really briefly, just to kind of get an idea of the diversity. We'll do Saccharomyces, so those are our budding yeasts. Alternaria, which is an important plant pathogen and even sometimes a human pathogen. Sordaria, which is a pretty famous genetic model organism, and then Xylaria. Okay, Sordaria is the one that you actually have a plate of it in your lab kit that you picked up. It's the plate that was already kind of bound up, and I think it says SF cross on it. If it says something different, let me know. But I remember writing SF cross. Okay, and that stands for Sordaria fimicola. Right. So Saccharomyces. This is our brewer's / baker's yeast. It does things a bit different. So this is kind of an exception to the life cycle. Looking at this generalized life cycle down here, what does it do differently from what I said was the rule in Ascomycetes?
It doesn't have an ascus?
It actually does. Pictured over here on the right.
Oh, okay.
Its entire body becomes an ascus, so that's different. Since it's single-celled.
Would it be there's no conidia?
It can actually do conidia if it's reproducing through mitosis. That's by definition a conidia right there. So you bud out, and then that yeast gets picked up by a bird and dropped somewhere. That counts as a spore. It's a reproductive structure produced by mitosis. So that's all that a conidium is. It's in the bottom right of this picture.
The diploid cell cycle?
Yeah. Saccharomyces are the weird ascomycetes in that they can be diploid for quite a while. Remember, I told you way back that they don't like to be diploid. Right? They're named the dikarya because they hang out in this dikaryotic stage where the nuclei don't fuse. And then as soon as they fuse, they go straight to meiosis. Okay. But, of course, there's the weird ones. And Saccharomyces are the weird ones out there. And they can actually stay diploid for quite a while. Okay. So they still have the same sort of mating structure. They can be haploid. They can reproduce through mitosis that way. But they can get together, mate, and then the two nuclei can come together and be diploid, and it can just stay in that cycle. It doesn't have to do meiosis right away like the rest of ascomycetes. Okay. So that's weird with them, but it gives them an advantage as a genetic organism because that's what animals do. And so we can actually use these to study like animal cells and not feel bad about it. Okay. We can also use this to study the evolution of sex. What's the advantage of being diploid? You know, so we take identical strains, grow them haploid, mate some of them and grow them diploid and don't let them undergo meiosis, and we can look at ecological advantages. Okay. These are also kind of weird in that they're okay with not having oxygen present, or at least having very low oxygen present. Okay. So you take flour and water and you make a dough with this particular ascomycete in there. You don't have a high oxygen content inside that bread dough. Okay, it's pretty sticky, not a lot of air getting in and out. Okay, so Saccharomyces are kind of freaky in that they're okay with that. Most fungi are going to be one or the other. Vast majority of fungi are aerobes, but Saccharomyces can just switch over to a new kind of respiration. It's not actually respiration, it's fermentation. Okay, so if you give Saccharomyces, put it in some water, mix in some sugar, and leave it open to the environment, it's going to do cellular respiration, get lots of ATP, grow, and basically produce vinegar for you. Okay. If you seal it off, put a lid on it and put a little gas escape so air can get in, or can't get in, but CO2 can get out, then you get ethanol. All right. So that's how we make vinegar, is we let oxygen in, and that's how we make wine, is we keep oxygen out. That's the only difference. Okay. Every grape you've ever bought has this like little dusty coating. Those are all Saccharomyces yeasts. Right. They're all over the place in nature, really sugar-rich environments.
So we could just rub a grape on one of our auger plates and...
Yep.
Okay.
And you've got yourself a Saccharomyces yeast. And chances are that it's a novel form and might be interesting to sequence and study. Yep.
All right.
There's tons of them, wild, all over the place. If you go into any flower, go into the nectar, there's going to be Saccharomyces yeast growing in there also. Yep. So you can take any flower and just squeeze the nectar onto an agar dish, and you've got Saccharomyces that way also. But yeah, the grape one's actually kind of fun. Just rub it on there. The original breads and like beers that people were making 10,000 years ago, right? They didn't have like a nice petri dish full of pure strain of Saccharomyces cerevisiae. What'd they do? To leaven their bread, they just left it out, or they actually mixed in some grape hulls with it. They didn't know why it worked, but it did.
I assume there's a lot of just like superstition of why it worked.
Yeah, exactly. And so this is the idea behind sourdough. Okay, you mix some flour and water, you let it sit outside for 3 or 4 days. It's going to pick up wild yeasts, and you close it off, let it start bubbling, and once it's working, you mix that in with your dough, and so you've got wild yeasts and bacteria and stuff growing, and so that's what's making your gas bubbles that way. The vast majority, they've done like sourdough sequencing projects, and that's just now kind of getting published. But by far, globally, no matter where you're making your sourdough, 75% of the organisms in there, at least it looks like, are going to be species of Saccharomyces yeasts. They're just all over the planet. You're breathing them in right now. Okay. But they do that kind of weird, they can hang out diploid. They can go through their life cycle diploid as well. Mating types, we don't have a male and a female here because they're the same size. They look exactly the same. It's all genetic. And so we call these A and alpha. So that's what you'll see in your text when they're talking about mating crosses and that kind of thing. A has to mate with alpha, and it's just a single locus in their genome, all right, on one of their chromosomes, and that determines their mating type. What is the point of being different mating types? Why not just let anybody mate with anybody? You look exactly the same. You're contributing the same amount of resources. What's the point?
Is it for tracking them through the reproduction cycle?
I mean, what's the point for the yeast? What does it get out of it? Having this mating type. It's essentially limiting who it can mate with, where there's essentially no need. They could all donate nuclei to each other and undergo karyogamy and meiosis. But they've got this little section on their genome that says yes or no to mating. Kind of goes back to the entire point of sex and meiosis in the first place. What's it good for?
Genetic variation.
Genetic variation. Okay, great. So let's say you're a haploid yeast and you undergo some mitosis and your cell gets bigger and then you bud off a little clone of yourself. Would you want to go mate with yourself?
No.
Nope. No variation there. I mean, this is a problem for humans too. In like really close societies, they run out of genetic variation and people are marrying their cousins and having kids, and then their kids are marrying cousins and having kids. It's a problem, right? Genetic diseases can build up. You want variation. So the mating type is actually there to promote variation and to keep you from breeding with yourself or something that's too close to you. Okay. However, they got to be weird about everything. They can switch their mating type after budding off. So this is maybe a bit counterintuitive, but it can divide, make a clone of itself, and then the mother cell can switch its mating type and immediately mate with its offspring under certain conditions. So maybe that's I have no idea what the benefit might be there. Maybe if you can't find a copy of yourself, some crossover is better than none. At least you're reshuffling your genes. Not really sure. But they got to be weirdos and they can do that. Okay. Another weird thing is we call it the Crabtree effect, and it's that these yeast sometimes seem to prefer ethanol fermentation, even though it sucks at generating ATP. All right, so regular respiration nets you at least 18 ATP, possibly up to 30, 32, some estimates 38. It depends on the organism and circumstances, but it's a lot more than going through glycolysis and fermentation, where you get a net 2 ATP. All right, so it seems like it'd be better to do respiration than fermentation, right? You get more energy for the same amount of work, same amount of resources, glucose molecules. But these yeasts will sit there in the presence of oxygen and ferment ethanol sometimes. Can you think of how that might be adaptive for them?
Well, is that just because of like they have, I don't know, like resources more abundantly in one way that makes them want to, I don't know, do that channel versus another? Or what guides that?
Okay, yeah, so actually when we studied this, because it doesn't make any logical sense why you would do the crappy version of energy production over the good one if air was present. It does depend on sugar concentration. Yeah. And so if you get over a certain amount of sugar in solution, they'll switch over to fermentation even with oxygen present.
Okay, thank you.
What's the benefit?
Is it less energy input on their end?
Exact same amount. 1 molecule of glucose. That's the resource they're contributing.
Is there less, I guess, so oxygen is destructive in a cell. Is there maybe less oxygen produced or less negative byproducts produced?
The byproduct of respiration is CO2, and that doesn't really hurt them. And so respiration is actually getting rid of oxygen, whereas fermentation isn't.
Do we know the answer?
We do. Yeah.
Okay.
Does it have to do with the symbioses with other organisms using the byproducts of fermentation?
Oh, you're real close. Not necessarily using the byproduct of fermentation, but being killed by it. Ethanol is the byproduct of fermentation. All right, so.
And ethanol inhibits like bacteria?
It does. And so this is why we use them to make beer and wine, is because they can tolerate ethanol pretty well. They're okay with it, but a lot of stuff isn't. And so if you're in a flower hanging out in a little nectar pool, this is a way to avoid competition for that sugar, is you start producing ethanol. Even though you get less energy out of it, you're the one that gets to use all that glucose that's sitting there. Okay, so it's a competitive advantage, them producing ethanol. So think of it kind of like chemical warfare that people just happen to take advantage of. Okay, they're producing ethanol and doing fermentation not because it's fun for them or they like getting less energy. It's because it keeps other stuff out of the syrupy sugar solution. Okay, so that's been a pretty active area of research, and we've used it kind of in a game theory scenario to look at trade-offs in the evolution of different biochemistries, that kind of thing. Like, why would you do something that seems disadvantageous? Well, let's look for where it might help. And so we can genetically engineer yeast. You can actually just go online and order a knockout version where they've knocked out any gene in their whole genome. So you could knock out part of the fermentation pathway, order that online for about $10 and experiment on yeast that can't do fermentation and see how they fare versus ones that can. Yeah, and they actually do better in a high sugar solution. Okay. So that's the Saccharomyces. They're really interesting organisms. I mean, they're boring to look at, obviously, but in the history of science, they've given us so much insight into how biology works, human biology as well. That's how we understand genetics and everything from them. Okay. And we've got Alternaria. Okay. These are important plant pathogens. As far as we know, not at least no known telemorph stages. Any day now, of course, we're going to find one, but for now, I'm just going to say, yeah, they are all asexual. Okay, so these are all over the place also. They require, a lot of these species require a living plant, and they are necrotrophs, meaning they've got to have the live plant, and then they'll kill it off and eat the dead body afterwards. Some of them are wood saprotrophs, so they'll just grow in dead wood, feeding on the cellulose, all that good stuff. And of course, here's an eyeball. So, as pleasant as that photo is, this is somebody that has an Alternaria infection in their eye. And this kind of gets back to that question of, well, some of these might have different names. You got to kind of look at that. But most of them are plant pathogens. Okay. Here you can see their conidia. They've got these crazy shapes with a bunch of lines through them. So what kind of spore is that? It's not an amerospore, because an amerospore is a whole entire single cell with no divisions. What do we call it? It's fragmented, and so we call it a phragmospore. Okay. But this is a single spore. It's just kind of broken up. It's got cell wall divisions. And now we come to Sordaria. Sordaria is the one that you're going to be looking at under your microscope. Okay. I've got a couple resources up on the webpage for Sordaria in the video section for this week. I want you to watch those to get an idea of why we're looking at Sordaria. Okay. But in essence, it's got really narrow asci. So the little sac is quite narrow. And you can see that the spores just really line up with each other, and there's no room for them to kind of move around. What this means is that we've got a record of how meiosis happened. Okay. So here we've got a cross between two different types. A wild type, which has dark black spores, and then a tan mutant, which has these tan spores. Okay, so we look at this, and here are the patterns that can happen if there is no crossover or if there is crossover. Because meiosis takes place, it's going to split into two after meiosis 1, and each of those is going to split into two after meiosis 2. And then there's a round of mitosis, and so each of those splits into two. They have nowhere to go. They can't jostle around each other. And so it's kind of neat that they're all packaged in here and we can see exactly how meiosis took place. Okay, so watch those videos. This is what you're going to be looking for under your microscope. All right, we'll talk about that in just a second. I want to show you Xylaria first before we talk about the lab exercise. It's our last group that we're going to cover from your readings. Xylaria. I don't want to say all, but all the ones I know of have this two-part life cycle, where half their life cycle they have to be inside of a leaf. We call that the endophytic part of their life cycle. And then they get dispersed out of the leaf, land on some wood or dead leaves or whatever, and then they eat that. Okay. So endophyte and saprotroph. Where is the sexual structure here, and what do we call it? I got an arrow pointing to it. What does that look like to you? Anyone?
Spitzenkorper?
Nope, that's the dark body at the at the tip of the hypha that regulates directional cell growth.
Is it what you call like the medulla, or is it just it looks like the hyphae itself?
All right, can you see my cursor moving around here? This structure right here is a cross section of this growing fungus. You may have seen these out in the woods. The common name is like dead man's fingers. Okay. Towards the top, they've got asexual reproduction going on. That's the white stuff. The black stuff here is sexual recombination. So up here in the cross section, we've got a sexual fruiting structure, and inside of it are asci. You can kind of see the spores in their little sacs hanging out, lining the surface of this cavity. And then the cavity opens up at a small little hole right at the top there, and that's where these asci are shot out. That's a parathecium, okay? Remember those terms because you'll see them in the reading. Apothecium, cleistothecium, parathecium. Okay. So this is parathecium here. Pictured up here in a drawing, you can see the asci are lining the inside of it. Okay, so that's sexual reproduction going on there.
Question.
Yes.
So for the picture that has the arrows pointed on it on the right side, the asexual, sexual, would you call the white part conidia and then the black part an ascocarp?
Yes.
Okay.
Yep. And that's because these get a bit funky because the black actually kind of moves up, taking over the white in these. They start out white and they slowly turn black from the bottom up. As they're becoming sexually mature, they start turning black. So this is kind of weird. The structure itself, the little finger sticking out of the dead wood, you couldn't call that an ascocarp necessarily because it also has conidia growing on it. Okay, so the ascocarp is just the lower half of that structure. Up at the top, that's not sexually fertile, at least not yet. It's still producing spores asexually. So they get around this whole like, should I do asexual or sexual by just doing both essentially at the same time. So they start out asexual and develop behind it. So they get the best of both worlds.
Do we have a term for this where it's that whole like finger-like structure? It's exposed. It's not like hyphae, right? Mm-hmm. Which normally we would just call the entire thing a fruiting body. Do we have a word for both of those together that are exposed?
Not that I've heard. There probably is one.
Okay.
In my 15 years as a mycologist, I've never heard it. I just hear people say the fruiting body of the xylaria, and then they say, oh yeah, I mean the sexual part, if you ask them. Yeah, or you can kind of pick it up from context. So there probably is a word. Of course, there's a word for everything in mycology. It gets kind of annoying, but I am not aware of it. If you call it the fruiting body, I'll know what you're talking about. If I ask a question about it, understand that it could be both asexual and sexual. Okay. Here, the top one, you can see that that's all sexual at this point. Okay, so at that point, you can comfortably call it an ascocarp. It's tricky trying to define what an individual is in a fungus, right? Because they can be many things at once. All right. So that's kind of a quick run through for the groups that you're going to be reading about. I just wanted you to see some pictures, have a chance to kind of get a feel for them before you read the denser textbook version of everything. Any questions about Ascomycetes? I want you to keep all this in mind as you go back through the slides. Think about how this fits into our picture of fungi. Okay. Barring any questions, let's talk about Sordaria. You've got one petri dish. Let me stop sharing my screen here. Okay. You got one petri dish labeled, I think, SF cross or SF mating cross. I put on one side a little culture of the tan, and on the other side a little culture of the wild type black spore producers. Okay. They've been sitting long enough that I bet right down the middle you're going to have sexual reproduction. Let me draw out what this might look like.
I'm looking at mine right now, and I got a thin line that's forming in between them.
Is it dark colored?
Yeah, they're. It's not a heavy line, but it. I mean, you can definitely tell that a line's going to form there.
Cool. That's what we're going for. Is basically like a little line between the two blocks of agar. All right, so one of them is tan, one is black. It doesn't matter which is which. What we're going to look at under a microscope are some of these ascocarps in the middle. Okay, so if you've got even a whisper of a line forming there, then you're good to go. It'll produce hundreds of ascocarps. Each little one kind of ascocarp kind of looks like that, like a little vase. Okay, and that's going to be sticking up off the surface of your agar. It's pretty tiny, so I expect you're going to need some small tweezers or like an X-Acto knife or something. But I want you to pluck one of those off, or even a few, if you have to scrape them off or whatever. Stick those into a tiny drop of water on your slide, put a cover slip on, and then give it a light smash. Okay. You really kind of need that smash to see what's going on. Let me see if I can find the photo. Yeah. Okay, so here's Sordaria again. You can see that they tried to indicate what they look like. All right. So looking from a top down, it's going to look like a little turd or something. Okay. Pluck some of those off, stick them on there and smash them, and that's how you get the asci to come out. All right. Don't smash it too hard. You're going to break your cover slip or whatever, or smash open the asci. So just give it like a little gentle smush, because we're trying to pop that parathecium open and get the asci out. We can't see through this. It's a nice melanized structure, so we can't see through it with the light microscope. So we need to smash it to get these things out. Okay. So for that assignment, I want you to get a photo of the surface of the agar with these things on it. So I want to see that ascocarp, that parathecium structure hanging out. And then I want a photo or two of the asci that are inside. Okay. And it should be a mix of black and tan spores inside each ascus. Okay. When you're taking those, make sure you
got a real quick question, sorry. So those ascocarps, those are the ones that are, we want to grab the ones that are in between the two agar pieces, right?
Yeah.
Okay. Because I have some that are above and below it, but very few that are in between.
Well, in between is going to be your best chance of getting a good mating cross.
So should we just wait until we get a couple more?
It only takes one or two. You should be okay. Yeah.
Okay.
So yeah, when you get in there, basically get a nice close-up picture. Your microscope will let you like just set it on top of your agar. Don't squish down too hard. Get a picture of one of these things up close. It'll kind of look like this, but from a top view. Okay. And then scrape or pluck a few of these off real carefully, stick them on a slide, and give them a little smush. So on your slide, it'll bust them open. Like that, and they'll kind of squirt out, and you'll have asci hanging out on your slide. All right. Now, go to the website and watch the videos about Neurospora crassa. There's just 2 videos on there, I think total of like 8 or 10 minutes before you do this, to give you an appreciation of what you're looking at. Okay, it kind of walks through a very famous experiment, the Beadle and Tatum experiments, you probably heard of. They use Neurospora. This is closely related to that. It's a little easier to work with in culture, so that's why I use it, but it gives you the same results. This particular species could have given the same data to Beadle and Tatum. Okay, that's Nobel Prize winning research right there from our dumpy little fungus. All right. So watch those videos and then get a close-up look at these asci and see if you can figure out the pattern that meiosis went through, whether there was crossover or not. Okay. What would it look like if there is not crossover? This is a photo, or not a photo, a drawing showing no crossover. Okay, we've got the black type and the tan type, no crossover. Then your spores are going to be arranged like this: 4 tan, and then 4 dark. There is crossing over. This is a chart showing you the different paths that meiosis took for crossing over, and it's going to show you the outcome of your different spore arrangements. Okay, it's got to be one of these or one of these. So look in there and see what you can find. People used this method before genotyping became a real thing. Okay, Beadle and Tatum was, I don't want to say a year here, but I think 1960s when they did their experiment. And since they knew how this thing grew and the fact that the spores had nowhere to go after the different rounds of meiosis and mitosis, they were able to basically like backtrack, and from counting thousands of these asci over and over and over, which I'm sure they had grad student servants do for them, they were able to basically get quantification of how often it happened and identify where on the chromosome the gene for spore coloration was. And they got it right, okay, long before we could do sequencing. Okay. So I'm not going to make you do that because that would involve counting thousands of these things and doing some statistics, but I want you to see it and get a close-up look at some asci. Okay. Questions?
Do you want us to turn that in this week?
Yeah, whenever.
Okay.
Should be able to get it done this week. If one of you has parathecia showing up on your plate, then you probably all do. So you should be ready to do it.
Okay.
Okay. And then the other assignment, how many Ascomycetes are there? I don't need like a 2-page paper or whatever, you know, just give me your estimate or your range of estimates and back it up with some peer-reviewed articles. Okay. Has everyone used Google
Say
Scholar?
that one more time. I wasn't sure about that. What that with the articles and what are you needing from us?
I'm looking for like a few sentences on how many Ascomycetes there are.
Okay.
But you backing up everything you say with articles from peer-reviewed literature. So I want like a works cited, you know, reference section underneath your short paragraph. If you're not familiar with Google Scholar, get familiar. Let me show you real quick. All right. So it's just scholar.google.com. What this does is return peer-reviewed articles instead of a bunch of crazy conspiracy sites or Facebook pages. Okay. So Google Scholar, go in there and start asking it questions. Number of ascomycetes. Maybe you don't find direct hits on that. Okay, you don't. All right, so you're gonna have to go in and look at the abstracts, find some of these that help you answer your question. And then I use a reference manager called Zotero. You may want to do the same. Zotero, your personal research assistant. It's free. It's got add-ins for Firefox or Google Chrome web browsers. And for example, I can open up my Zotero here. So this is Zotero, it keeps track of stuff. Make a new little folder. And I'm gonna say, come on now. I don't know why it's being a little jerk to me. But I'm going to save this to Zotero just by clicking a single button. So no mucking about with trying to format your references, no worrying about getting them in correct alphabetical order or any of that. Zotero takes care of all of that for you. You just go in, find the papers you want to cite, save them, and look at that. It keeps them for me. I can create a bibliography from those, pick whatever format I want. I'll do APA 7th edition, copy to my clipboard. And then open up Microsoft Word and I can just paste it in. Okay. So that's how I would approach this, is using Google Scholar and Zotero to let you kind of keep track of what your sources were. Okay. Make sure you are looking at the website resources that I've put up for you. We've got an interesting paper on the species concept in fungi. If you're interested in why we do things the way we do so stupidly, and kind of where we're going with that and how we're trying to fix it, that's a cool paper. We've got a whole web page on how an ascus is formed, paper on anamorphs and teleomorphs, and then I gave you links to Saccharomyces, Sordaria, Alternaria, and Xylaria, so you can get a look at those. And then the videos of Neurospora crassa and Sordaria crossing over are going to be really helpful before you start smashing your parathecia. So go through those, see what else. Okay, that's all I have for you. There's no journal article for your reading this time. It's just textbook chapters. Any questions?
Just wondering when you're going to get that quiz up for us to do for this next chapter readings.
Today. Yeah.
Okay. All right. Appreciate it. Thanks.
Mm-hmm.
When is the ascomycetes assignment due?
I don't know.
By the end of the semester, he said.
Yeah, by the end of the semester.
Oh, I mean the one about how many there are.
Same.
Oh, no deadline?
Yeah, it may take you, more than 30 minutes to an hour to get this done, especially if you're like trying to figure out how to use Zotero, which you should. It's going to save your life as a student to have a good reference manager. And so I'm factoring that in. And yeah, end of semester. The sooner the better. I mean, get it out of the way before you start getting midterms and stuff in other classes.
Yeah, for sure. Thank you.
Mm-hmm. If you've had one of my classes before, you will have seen how I operate with like due dates. The faster you do it, the better it is for you. If you wait until the end, that's kind of a your problem thing. I don't worry about that.
Cool.
All right. Well, that's all I had for you today. So we will chat about the reading on Thursday. And then after that, we're moving on to basidiomycetes, and that'll be the last group that we look at before we start delving into what fungi are actually doing out in the environment. All right.
Thanks, Dr. Zahn.
I will see you all next time.
See ya.